Hello student, continuation to microbial culture. Today we will discuss about the culture procedures. So what is the meaning of culture procedures? So it is now culture procedure is the various techniques used to do the process of culturing of microorganism, and hence these culture procedures are depend upon the three techniques or three fundamental rules we are applying here for the culture purposes or for the culture procedures. First one is the sterilization. This meaning of sterilization is what? To make the surface sterile, that is germ free, to make germs free. You know, these bacteria, fungi and other unknown microorganisms, they stick to the surface of the fermentation, to the surface of fermenter, to the surface of the equipment what we are taking for the culture process. So basic need is the sterilization. Now this sterilization, if we are doing in the laboratory, so normally we will be using the buffer flask. Normally this is the buffer flask, and it is normally ranges from 100 ml to 1000 ml. This is for the short scale production that we are doing in the laboratory. So how we sterilize these buffers or the equipment what we are using in the laboratory are sterilized by the autoclave. It means by the process of autoclaving we are sterilizing the surfaces, and here what we are using the 15 pounds, that is the side pressure, for 15 to 20 minutes. So sterilization in the laboratory is done for 15 to 20 minutes under the pressure 15. But for the large scale production, that is when we are using a fermenter. You know that fermenter are so big metallic vessels where we are adding the culture material, microorganism, nutrient medium, etc. in appropriate proportion, but it is more than 1000 liter. So here autoclaving is not possible. Then what we are using here? Normally we are using the steam therapy, that is the steam sterilization process, and therefore steam is universally accepted for the large scale production. So this is the steam, and steam is largely used for the large scale production or large fermentation for the time period 20 minutes and under pressure 15. So 15 under pressure, here we are autoclaving also, that is the 15 pressure, under the 15 side pressure for 15 to 20 minutes, and these large fermenters or for the large scale productions we are doing 20 for the 20 minutes under the pressure 15, but we are using here the steam, and this steam is sparged, that is it is scattered among in the fermenter. Now here we are also in the nutrient medium, so nutrient medium must be also clean, nutrient medium must be also sterilized. So we are using two conditions here. If medium is sterilized separate, then fermenter must be sterilized before sterile medium is added. It means suppose in the laboratory we are doing this culture purpose, so nutrient medium is added separately. So before adding to fermenter, that medium will be sterilized. But the question is that here what will happen for the large scale production or in the large metallic vessels that we call the fermenter. So here in situ sterilization is done. So there is two kind of sterilization, in situ and ex situ. In situ means within the fermenter, and ex situ means outside the fermenter that we are doing in the laboratory. So in situ is sterilized in the fermenter. Now before going to that, second part is aeration. Actually this cultural procedure requires three phases: sterilization, aeration and mixing. So aeration is air is also required for the culture of the microorganism. So what we are using, how how we are doing the aeration? The air used in fermenter is filtered by the filter sterilization technique. So what is this role of this air? This air that we call oxygen is sparged with the star. There is mechanical stirrer. Now this is we are normally using the stirrer like this. Suppose this is the buffer flask, so we are now normally using with the help of this glass rod. But here it is not doing so. In the laboratory, the culture procedure is different as the large scale production. So this mixing, there is also known as sec culture. We are putting flask on the shackles. Now they normally this move like that. So due to this movement, what happened? The air is get mixing properly. But the question is that if we are using the large scale production in the fermenter, what we are doing here? We are using here mechanical stirrer and forced aeration done for mixing oxygen. So this mixing is not proper mixing of oxygen or aeration. What happened? There is creation of the bubble, so that create problem in the culturing of the microorganism. So aeration and mixing is done. So this is about the culture procedure, and the culture procedure is under head of the three important stage, that is sterilization, aeration and mixing. Literal meaning of sterilization is to make the germ-free surface. Aeration, mixing of addition of oxygen or air, and mixing, that is these oxygen or air get mixed to each of the cells, which is let down in the culture medium or in the buffer flask or in the fermentation. So this is about the culture process. Thanks for watching.